For the final talk of the day, which is by Chris Greenman. Chris is at the University of East Anglia and also has, I believe, it's a joint appointment with TGAC, the sequencing centre in Norwich. And Chris comes from a background starting out in physics and then moving out into industry. Back into the Sanger Institute to do cancer genomics on the strength of his statistical skills, which he'd acquired basically whilst out there in the real world. And from there, he went on to, by the way, it's very interesting stuff he did while he was with the cancer genomics group. And so now he's at UEA, and he's going to tell us about some of the things he's been doing there. Thank you.
Well, thanks for the invitation and the opportunity to speak. So I'm going to talk about the evolution of viruses, which, as Nico mentioned, it's a bit of a stretch, but you can kind of think of it as a simple metagenomics problem, in the sense that you've got a really good reference that you want to use, and you can think of it as the diversity is quite low, although for a single species the diversity is actually quite high. So in terms of the problem, what we'd like to be able to do is to take the kind of virus sequencing data and quantify the haplotypes. And by that I mean kind of two things. First of all, identify what haplotypes there are, and secondly, say how much of it there is in the population. So I'm going to kind of use two sources of information. One is kind of like local haplotype structure, where, for example, you see two mutations on one paired read. I'm also going to try and point out that the mutation prevalence itself, the frequency of the mutation, also contains quite a lot of information that you can use in some circumstances. And then from that, we're going to try and get the evolutionary structure of the underlying viral population. Now, when I first tried to do this, I tried to do it with trees, which worked in some cases, but actually there's a lot of recombination that goes on, and so we had to turn to networks. The other thing I should say about this, I guess, if you're thinking about a metagenome problem, is that this is time series data that we've got. Now, admittedly, viruses change quite quickly, but you can kind of control the environment quite nicely and control the way that the virus moves from animal to animal. So this is also time series data. So what is the flu virus? Well, it's an RNA virus, which means a single-stranded RNA virus as well, which means it mutates really quickly. I mean, there's no repair mechanisms. I mean, the thought is it mutates once every 100,000 bases or something, and flu itself is about 10,000 bases long. So every 10 new viruses, you're going to have some kind of new mutation that pops up. So it's going to cause quite a bit of diversity. So I'm going to mention a couple of data sets. I'm going to kick off with some nice data from a little while ago. And this is actually Sanger data. It's not next generation sequencing data. So I think what my understanding is, they kind of diluted the flu so that on average you've got one viral particle in each well, and then they Sanger sequence it. So each horizontal line you can think of as one viral particle, and this is the genomic position of one of the segments in the flu virus. And so what we can see is we get this mutation that's piling up in the same place. And the thing to note is that if you take any single row, you either get a blue or you get a green. They're kind of mutually exclusive in this case. They never occur in the same row. So for this particular example, we in terms of a tree at least, you can think of that as two separate branches where you've got blue on one branch and green on the other branch. But continuing from that, we can kind of count the number of samples that we have, get an estimate of how much of the blue there is and how much of the green there is, and it gives us some idea of the population size. Now the total population is 100, and that's kind of splitting into these three ratios. So what we can kind of think of as this kind of mutation prevalence, it's kind of like a flow that passes through this tree. So it's conserved, the 100 splits into the three different terms, but they add up to 100. So we got this kind of conserved network flow, if you like. So the type of experiments we're talking about that generate this data, they're kind of transmission experiments. So you start off by growing up flu in an inoculum. They talk about growing it in an egg. I'm not really sure what they mean by an egg, but it grows in something. And then they pass this to two animals. So these two animals get the inoculum, they're put in the same room as these two animals, then after a while those two disappear, those two go in a room with those two animals, those two disappear, and you get this kind of transmission chain that occurs. And you take daily samples as well. So for each of these pigs you have 4 or 5 samples on a kind of daily basis to build up a profile of what the data is like. So I'm going to mention a couple of data sets. The one we just saw was data from pigs, and that's the Sanger data. We will have a look at some next generation data as well, which is from a chain of pony infections. So if we turn to the pig data for the moment, then what we had was these 2 separate branches with the 2 mutations. But this is actually what happens on day 1. We can only see one of those 2 mutations there. We can only really see the blue one. So it could be that the green one is there at a very low prevalence, or it could be that it's just not formed yet. Now in some sense it doesn't matter. What we can do is we can still have this same tree structure, and we can just say that the prevalence of this is very high and the prevalence of this is either 0 or negligible. So we can keep the same kind of topological structure, and it's actually these prevalences that are changing. So when we go to the next day, the blue one's not so dominant, the green one starts to pick up a bit, and so this rate has gone down and that rate has gone up, but it's the same topology. So I mentioned the prevalence, and there's a desire to try and use it in a quantitative manner. So one of the things we did to see if we really could use it in a quantitative manner was to kind of do separate libraries. So this is next generation data now. So this is a high-seq run, this is an older MySeq run, which is a little bit ropier. But the point is that the prevalence of both of these mutations is actually pretty stable. So you can use this percentage prevalence as a kind of an indicator of the population size of the mutation. So this is just a different day. We've got the same mutation cropping up, and then when you go from MySeq to HiSeq, you've got a similar kind of height cropping up, which is nice. So I talked about the prevalence being useful. So here's some sample where we've got these 5 mutations that have quite high prevalence. So we've got the ones I've circled in red. So they've got a prevalence of about 55%. And the blue ones have a prevalence a bit higher, about 70%. So I'm not saying anything about kind of the haplotype blocks, but what I would say is they can't be on separate branches, because if this branch has 55 and this branch has 70, they add up to more than 100%, which doesn't fit. We've got some other mutations at a similar level there. So if we assume a tree kind of structure, the only thing we can really have is that red must be a descendant of blue with a slightly lower prevalence. So red is at 55, and then blue is the sum of those two, which is 70. So this is same animal, a day later. And what we find is that the blue mutation still has quite a high prevalence, but this red stuff has kind of died away somewhat. So in terms of this tree, what we can say is that this leaf has gone down in prevalence, maybe because of negative selection, I don't know, whereas this one has been preferably selected in some manner, and it's gone up from 15 to 60. Again, so we've got the same topology, different prevalences. So that's one way of using the prevalence kind of information, but there's also kind of local haplotype information as well. So these are 3 mutations we found on segment 4, and they happen to lie in such a manner that if you have a paired read, you can actually see all 3 of these mutations at once. So then you could look at the table of different mutation types to see what they do. What we can note is that we can have there's 4 rows basically that have quite high numbers, and the other rows are very low. So our interpretation of this is that there's 4 genotypes that are actually in the system, And these lower numbers, like the 4s, the 3s and 1s, are just kind of artifacts we can ignore. So in terms of a tree, that tells us we've got some kind of tree-like structure involving these 3 genotypes. So we've got the local haplotype structure, which helps us form a tree. So in general, the problem is we're trying to reconstruct some kind of evolution tree, ignoring networks for the moment, based on this kind of information. So we've got separated segments. We may find that our reads are not long enough to actually bridge gaps between mutations. So we've got this kind of clustered pieces of information available. So here we've got 2 mutations that are bridged by a read. That gives us some kind of table from which we can construct a tree. Same here, we've got a single mutation. It's not really linked to any of the others very much. So that gives us a tree. And then we've got the 3 mutations here that are linked. That gives us a bigger set of things to look at. Again, we can construct a tree. So what we have are 3 trees, but what we actually want is a kind of super tree that contains all of these trees as subtrees, if you like.
That was quick.
Oh, I have jumped along a lot, haven't I? Okay, so those are our 3 trees from which we want to get the super tree. Now for trees, the prevalence is actually quite helpful. So what we can do is we've got 6 mutations scattered across these 3 trees, and each of those mutations, we know its prevalence. We know how much of it is occurring in the population. So what we can do is we can order those mutations by the prevalence itself. And because of this kind of conservation of the flow through the tree, what we kind of have to have is that the higher prevalences have to be above lower prevalences, basically. So what we can do is we can build up the tree iteratively. So first of all, we've got M5, which is a prevalence of 80, so we just stick that on. Now the next mutation is M2, which has a prevalence of 34. Now we can't put it here because 34 is too big. There's only 20 available, so it has to go under M5. So we stick it there. And then next is M3, which has a prevalence of 31. Now that has 2 possible positions here. It can't go there. So we're not going to get unique answers from this. We're going to get several trees that pop out. So one of the choices, we've stuck it in, and then we keep going. That's the same tree. And now we want to stick in M1, which is a prevalence of 18, which seems to be able to go here, here, or here. But we know that M1 has to be independent of M2, so it can't stick underneath this one, so it only actually gives us 2 choices. So we've kind of got to respect the tree structures that we've found so far. So we stick that in place, and then we keep working our way through. So we've got M4 goes in next. M4 is a prevalence of 15. Now there's various places M4 can go, but from this one, M4 can't be under M5. So again, it limits the number of places that we can place the mutation. So we keep doing this, and what you finally get is a tree. Well, you get several trees that are consistent with the data that's observed. And the one solution I selected was the same as the one that we start with, but you can get multiple solutions. So what you kind of find in general is that when mutations have high prevalence, they're kind of quite high in the tree. We can position those quite well, whereas the mutations with lower prevalences, they tend to be able to be placed all over the tree, a bit lower down, and you're less certain as to what happens then. But the other thing we have is time series data. So we do this process for 1 day's data, and it gives us a set of trees, and then we do that for each set of data that we have. And because we think the structure of the tree has to be the same, we're looking for trees that are consistently across our time series data. And that kind of reduces the solution space quite nicely. So segment 6 was kind of the biggest tree that we found. It's got all separated branches. It's actually not interesting as a tree. It looks like we've got two mutations that are subsets of each other, but they're actually two mutations that are very close with very similar prevalences, so they're probably formed at exactly the same time, actually. Part of the reason we can't find kind of quite big trees when we're doing this is because there's kind of quite a lot of recombination that goes on. And we kind of have to turn to network-like solutions. So what kind of recombination do we have? Well, we can have cases where two viruses with different kind of genotype profiles enter a single cell, and they kind of get mixed up, and that's called reassortment. You can also have kind of this template switching that goes on within segments as well. So you can have recombination occurring all over the place. So here's a typical kind of data set where we think we've got recombination going on. So this time we've just got two mutations that are connected by paired reads. So we've got four possible genotypes. Now if this was tree-like, three of those should be real and one of them should be false. So we should have one row that's got really low values. And the reality is we don't. All four of those are very high. So there's definitely all four genotypes going on. So there's definitely some kind of recombination that's going on within that segment. Now you can construct phylogenetic networks, but we'd like to use the prevalence. So it's a bit more natural to think of these in terms of recombination networks. So you start off with the root, no mutations, then we've got a mutation on each branch, and then these two get recombined, and then this population has both mutations. So that's one kind of possible explanation as to what's going on in this table. So we tried a kind of similar approach. So we've got some kind of mutation data set where we've got some mutations that are grouped into a cluster by paired reads. This is separated a bit, so it's a separate cluster. So for each of these, we can construct either a tree or a network. And then what we want to do is to construct some overall picture of what we think is going on to the entire data set. Unfortunately, this is a bit harder than it is for trees. For trees, you have this nice conservation that happened. But here we've got mutation 1 with a prevalence of 69, which is higher than 29, but it's downstream of the tree. And that's because when it's recombined, M4 is not passed through that node. So that's why there's zeros at the end here. So you don't have such a nice conservation that occurs. So we did a kind of brute force approach, and we looked for all kind of networks that are consistent with these pair of these, and it gives hundreds of networks that you kind of then have to decide which are possible explanations or not. So then we come to a similar question: can we use the prevalence of some manner to help us decide what's going on, and does the time series help? So the answer is kind of yes in both cases. So if imagine, so these are the prevalences of each of these clones, but we don't actually kind of know what they are. We know the prevalences of the mutations. So we got the recombined node. So I just said I don't know what that is. That's A. Whereas mutation 2 has a prevalence of 22. And then we find M3 has a prevalence of 63, and that gets passed to all three of those nodes. So we know that the sum of those three has to be equal to 63, so we get this kind of linear term here. So you can kind of compile a set of linear terms here that are the population sizes of each of those clones. But the thing is, these all have to be positive because it's an estimate of population size. So what we find is that A has to be bigger than 18, A has to be less than 47, A has to be less than 41, so that's fine. So we've got a solution set, so it's valid, so that's a valid network. And we can check this, it's just a simplex method. So this is a different network that's kind of consistent with the two subnetworks that we start with. But this time we've got a different set of linear arrangements here. And this time we've got a term that's 7 minus A, which means that A has to be less than 7, whereas we've got a term A minus 10 here, which means that A has to be bigger than 10. So there's no solution to this particular network, so that network gets thrown away. So that kind of reduces the set of possible networks. So we throw those away. One of the unfortunate things with the networks at the moment is I kind of end up with solution ranges for the prevalences of these nodes rather than an exact estimate. So you don't get perfect information about what the prevalence is when you have a valid network. So we tried running this over the data, so segment 4 was probably the most interesting. So we tried fitting a tree, that didn't work. We tried it with one recombination event, that didn't work. And so we tried 2, and that's when we start to get solutions out. Now if you do 1 day's data, you get about 300 recombination networks. But we've got this kind of time series of animal infections. And when we do that, that number kind of drops down quite a lot, and we've only got 22 recombination networks to worry about. So the solution space has shrunk a bit. And they correspond to 4 different kind of phylogenetic networks, 4 different clone sets, if you like. So we're not getting perfect prediction, but it does narrow down the search a bit. Now these are the different types of clones that pop out. So for example, this is a clone that contains these 3 mutations. Now there's 22 networks it occurred in that we found, and this clone occurs in all of them. So these are kind of the most confident set of clones that we have. So though we don't quite know what the network necessarily is, what we know is that there was 2 recombination events, and we also know that this kind of tree structure corresponding to this is sitting inside the solution space somehow. So you can visualize these things. There's this extended Newick representation. So we put that in Dendroscope, which gives you some plots that you can look at. Okay, so that was recombination within a segment. So the other way you can get recombination is reassortment, where segments mix up. Now we don't really have any kind of linkage information because you can't sequence through several segments. So the only thing you really have is prevalence information. So these are 3 mutations in 3 different segments, and I picked out 3 different days that we sampled data from, and these are the prevalences. So if we assume a tree-like structure, we can kind of argue our way through this. We've got 57 is less than 68.6. So blue has to descend from green if it's a tree. We go to a different day, we've kind of got red is less than green, which is this one here, and they add up to more than 100, so they can't sit on separate branches. So if we fit that into a tree, it's got to kind of be that structure. But of course we're looking for one tree, so if we try and find trees that have these as subtrees, there's only 3 possibilities. We've got green as the parent, and red and blue are either separated or descending from each other somehow. So then if we work our way through the data, we've got blue plus red is bigger than green on that day. So those 2 together are bigger than that one, which can't be possible because you've got this kind of conservation of prevalence. So that tree's no good, we can throw that one away. So that one goes. Then we've got blue is bigger than red on that day. So this tree can't be possible because blue's descending from red. That should be a smaller population. So we throw that one away. And then lastly, we've got 21.7. So blue is less than 34.1, which is inconsistent with this tree. So that tree's not possible as well. So by kind of looking at the table and seeing what trees are possible with the prevalence, we can kind of decide that there's no trees that are possible with this set of data. So there must be some reassortment that goes on between the segments. So when you run the algorithm over the data, we find we get 16 recombination networks, 3 phylogenetic networks. Some of the clones come out in all of them, so we've got 709 is always there on its own. It's always mixed up with the other 2 mutations, but then the other clones are a bit unsure. So what we do know when we look through the data set is that segment 4 is definitely recombining with one of the other segments, but I can't actually tell which one it is. but I know it's happening. So in summary, I'd say there's two types of information. With paired reads, you've got this haplotype information that kind of links the mutations together. But there's also the prevalence, the kind of the height of the mutations, which gives you quite a lot of extra information. The other thing I would say is that, I've not really talked about this, but to start with, we looked at data where it was filtered for where the insert sizes were quite controlled, but you want to kind of bridge as many widths as possible. So we started, so although the average insert size was, I don't know, 500 bases or something, we started to pull out the bits of data where the insert size had grown and the insert size had shrunk, just to get a lot better kind of connective information. So we can detect reassortment. So I was kind of expecting that reassortment would happen. I must admit, I didn't think there would be any recombination between the segments itself, but when you look through the data, it's happening all over the place, actually. And you can start to build up a picture of what you think is happening in terms of evolution, for these viruses at least, when you've got time series data and the prevalences are kind of fluctuating enough to actually allow you to build up a picture. So this was work with Pablo Muccia, who's a vet in Glasgow now, and Donna Tiem did most of the work. And I'll stop there and take any questions.
Thank you very much, Chris.
Yeah, I have a similar expectation than you had initially. I think there's been few, I don't know, reports of recombination within segments of flu, though it's more common for HIV viruses, for instance. And in your presentation, it seems like you, in your example, there was just one mutation that couldn't be explained with this tree topology.
Right.
So did you consider that there are reverse mutations with some positions such as variable?
Yeah, that's certainly possible. So we have kind of assumed the infinite sites model where it kind of says that you can't have the same mutation twice. And of course with viruses, you've only got 10,000 sites, so it kind of is possible. And if selection's playing quite a strong role, and you've got a site that's of very keen interest, then again it becomes even more plausible that they can occur at the same time. So I haven't ruled that out or cross-checked or anything.
So it would be interesting to see if it's really multiple mutations or just individual ones that always conflict with a tree for 1 second.
Yeah.
Does time series data always help, or is it only when I don't know? I can imagine that sometimes you will have several samples and they will be so similar that the space of your solutions not actually.
I think that's right.
How often does that happen?
So I think equilibrium is the key. So part of the, I don't know how much equilibrium there is, because it kind of, it starts to build up, then the antibodies try and kill it down, then it goes to a different animal, which is different again. So I suspect there's not that much equilibrium, but what's certainly true is that when they grow it up in an egg to begin with, that's a very different environment to animals. So it kind of picks up fitness for that egg. And then when it goes into animals, there's probably a bigger transition at that point. So there's a greater change of what's going on. But if you do have mutations where mutations kind of go to fixation from one animal to another, then you are going to get some kind of growth that crops up. But you're right, you're kind of looking for those changes to occur to give you more information.
On the same vein, have you actually repeated this experiment in different host cells to see whether or not you have different rates of mutation at different points?
No, I mean that kind of thing would be quite interesting to do. So I've not really thought about the prevalences in terms of selection or anything like this. And there are experiments you can do because they've got antibodies against this and that. So the idea would be that you would kind of grow it up and then put the antibody in and see what changes. So there's lots of nice experiments that could be done in that fashion, but we've not looked at them.
I'm just curious about one thing. I don't know if I understood this right, but it looked to me there was only 2 sites there that seemed to be undergoing mutation, at least only 2 that you were analysing. Is it? Did I get that straight? Or?
No, you can kind of analyse as many as you like. if you analyse lots of mutations, the one thing you're going to run into is that the size of tree space gets enormous, and we're not being very efficient about that. So that's kind of one limitation. So what we kind of ended up doing was, it was a bit crude. We just took mutations that had a prevalence above 10%, I think, so that we were pretty confident they were real. But you,
So this particular one you started out with, where it seemed that you either had the green or the red, I didn't really understand that, but it looked to me like these were very correlated mutations. Was that?
Oh, I see. Yeah, so we were searching to see if we could find reassortment. So you do need mutations that create the kind of conflicts with the prevalence. So those were the 3 mutations that allowed us to actually see the reassortment. I mean, you can try picking 3 mutations with low prevalence, but they're probably not going to have enough information to cause a conflict, so you won't see the reassortment. Yeah.
I'm tempted to, well, I will ask it. I mean, this looks very similar to some of the work we did in cancer genomes. And so, I mean, that was much more statistical. This is more sort of algorithmic. But is there a similar way? Are you thinking about things in a similar sort of way?
I must admit I haven't personally, but people are thinking about these things. You can start to build up these networks, and it's quite difficult to automate it actually, because you end up having to kind of manually create a lot of the cancer data to work out where the copy number changes here, why this branch is doing that. It's quite difficult to automate the process.
In fact, this was more or less done by hand.
Well, kind of. The nice thing about viruses is that you don't really have copy number alterations, you don't have rearrangements. So in many ways this is a simpler model of cancer, if you like, because they've got a lot of similarities in the way that clones grow and separate, etc.
So we have no further questions. Thank you very much, Chris.